Hello, in this video we are going to cover the concept of getting information from DNA and converting that into RNA, and then getting that information from RNA and converting it into protein. And so essentially what we're going to be talking about today is what is the point of DNA. I know in a few of my videos I've mentioned that DNA is kind of the information of the cell. It is the thing that carries all of the genetic material or genes, but what does that even mean? What are we even talking about? So today we're actually going to talk about what this information is and how it translates into RNA and protein. So this whole concept of DNA to RNA to protein is called the central dogma of biology, and that is because all living things go through this process. So every living thing on Earth has DNA that contains information that is going to be converted to RNA and then later on used to make protein. So when I think of DNA, I think of a cookbook, because a cookbook contains many recipes to make different types of food. So I could have a cookbook that has a bunch of recipes on how to make different types of cakes, different types of entrees, soups, salads, whatever. And so think of it as like the ultimate cookbook. It contains the recipes for every protein that cell is going to need. And those recipes, we call them genes. So a gene is a recipe to make a specific protein. And again, the DNA of a cell contains all of the genes, all of the recipes. Now, RNA is just a copy of one single gene or one single recipe. So I like to think of DNA as like the most precious cookbook that your family has. Let's say it's a cookbook that's been passed down for generations. So it was your grandma's cookbook. Now let's say a friend texts you and says, hey, your grandma's chocolate cake recipe is super good. Can I have it? Or can I have the recipe? Now, you're not going to give your friend the entire cookbook if they just want to make one type of food from that cookbook, especially if it's a very precious thing like a family cookbook. And so what you would probably do is either take a picture or make a photocopy or even just write it down by hand, the recipe for that one chocolate cake recipe that your friend needs, rather than giving them the entire cookbook. And so that's kind of how the cell is. It doesn't want to use its RNA. The RNA is very precious to the cell. The DNA is very precious to the cell. And so it doesn't want to just use the DNA directly. What it wants to do is make a copy of one recipe, the protein that it wants to make, so the recipe for one protein, and it's going to make one copy of that recipe into a RNA molecule. And so think of RNA as just one single recipe to make one single protein. And then that RNA is going to be taken into the kitchen, and we're going to make the final product, which is the protein, in this case the cake or whatever it is that you are making. Now, the process of converting DNA into RNA, or using the information to make a copy, this process is called transcription. So making a copy of a recipe into RNA is called transcription. And then using that RNA to actually make the protein is called translation. So those are the two main steps. So DNA to RNA is called transcription, and RNA to protein is called translation. And so for the remainder of this video, we're going to briefly go over what happens in both transcription and translation. I'm going to go over it in the most basic steps. Obviously, they're much more complicated than what I'm about to explain, but this is just an intro to those two concepts. So let's start with transcription. Transcription starts with this enzyme called RNA polymerase. RNA polymerase is the enzyme that converts DNA to RNA. And so what it does is it will find the promoter, which is the beginning of a gene. So kind of like when you're using a cookbook, you usually go to the table of contents and then it says, okay, chocolate cake is on page 5. And so you go to page 5, and usually in big bold letters at the top of that page, it should say chocolate cake. Right? And so that's how you know that is the beginning of your recipe. So for cells, they know that the beginning of a gene is starting because there's this promoter region. So the RNA polymerase finds the promoter for the specific gene that it wants to copy, and then it's going to run along the gene and make a copy, and again, that copy is going to be in the form of RNA. And what it's copying specifically is the template strand. So remember that DNA is double-stranded, and there is usually one strand that is what we call the template strand, and the other strand that we call the non-template strand. So RNA polymerase is going to be, quote-unquote, reading the template strand to make our RNA molecule. Now, how does it make the RNA molecule? It's going to do very similar to what our DNA polymerase does during DNA replication. It's going to use base pairing rules. And so down here, imagine if we were zooming in on this diagram here. So we're starting over here, and what it's going to do is basically look at the letters, and it says, okay, I see an A, I'm going to add a U. Now, why a U? Remember, we're making RNA, so every time you want to add a T, you're going to add a U instead. So it sees an A, it's going to add a U. If it sees a T, it's going to add an A. And it's basically using base pairing rules to make this RNA molecule. Another thing I want to note is that this is happening in the 5' to 3' direction, very similar to DNA replication. So that is transcription in a nutshell. Now, once we have made our RNA molecule, again, this is the copy of one recipe, we're going to go to translation. Now, in translation, we're going to be using the ribosome. So think of the ribosome as the chef. It is responsible for actually making the protein. So it's going to, quote-unquote, read the RNA and make the protein. Just like when you're cooking, you're going to read the recipe and you're going to make the food. So what the ribosome does is it looks at the RNA and it looks at the actual sequence in the RNA and it reads it 3 letters at a time. And so that's kind of the word. So, you know, in a recipe you have like a word, and the word is made up of, for example, 3 letters or 4 letters. So milk, M-I-L-K, is a 4-letter word. Egg is a 3-letter word. So in biology, all words are in 3 letters, and these 3 letters we call the codons. So the ribosome is going to read it 3 letters, and then the next 3 letters, and then the next 3 letters, and those are the codons. And each codon represents a specific amino acid. Remember, we are making proteins. Proteins are made of amino acids. Those are the ingredients. And so how does the ribosome know which ingredients to add? It's literally going to read the RNA 3 letters at a time, and each 3 letters represents a specific ingredient, aka amino acid. And so down here is the actual codon chart. So what this means is if the ribosome sees a UUU, it's going to add the phenylalanine amino acid. If the ribosome sees CCU, it's going to add the proline amino acid. So it basically just translates it from 3 letters to a single amino acid. Okay, and so once that happens, it's going to add one amino acid after another. The ribosome binds them together using a peptide bond, and we have our primary sequence for our amino acid or protein. And that is essentially what's happening in this diagram. So it's reading the first 3 letters, it adds the methionine, it reads the next 3 letters, it adds the phenylalanine, etc., adding them to each other. Now, obviously there's a lot more going on here, but this is just kind of the overview of what translation is. Hopefully this video was helpful, and I will see you in the next video.